Welcome to our exploration of bioremediation, nature's answer to pollution. In this presentation, we'll look at how living organisms can clean contaminated land and water, why this approach matters, and what tools scientists use to harness microbial power. As you listen, imagine ecosystems being restored through biological processes rather than heavy machinery and chemicals. This chapter frames the pollution crisis we face today. Industrial activity, agriculture, and improper waste disposal have pushed soils and waterways to dangerous levels, creating long-term threats to ecosystems and human health. In the next few slides, we'll examine the scope of contamination and why conventional cleanup methods often struggle to keep up. Global pollution is reaching a tipping point, with a majority of soil contamination tied to mining, agriculture, and industry. Toxic pollutants like heavy metals, pesticides, plastics, and hydrocarbons are pervasive, and the resulting health impacts, from mercury poisoning to other chronic diseases, show why scalable, sustainable solutions are urgently needed. Industrial pollution has left many rivers, lakes, and soils critically contaminated, and the images here reflect landscapes under siege. These polluted systems disrupt food webs, reduce biodiversity, and create long-term risks for nearby communities, making remediation not just an environmental priority, but a social and public health one as well. Persistent organic pollutants, or PAPs, are a stealthy threat because they resist natural breakdown and build up in fatty tissues. As these chemicals move up food chains, they become more concentrated through biomagnification, threatening wildlife and humans alike, and highlighting the limitations of some traditional remediation methods. The human and environmental toll of contamination is wide-reaching. Toxic metals accumulate in vital organs, causing cancer and neurological damage. Soil pollution reduces crop yields and food quality, and illegal dumping and unregulated effluents amplify the problem across borders, creating both local and global crises. Chapter 2 shifts to the science behind bioremediation, where microbes become our primary allies. This section explains how bacteria, fungi, and algae are harnessed to transform pollutants into harmless byproducts, and sets the stage for understanding the biological mechanisms that make remediation possible. Bioremediation uses living organisms, mainly microbes, to degrade or neutralize pollutants, a concept that gained traction during oil spill research in the 1970s. At its core, it's about transformation, converting toxic compounds into carbon dioxide, water, and biomass. So the contamination is not just moved, but actually broken down. Our microbial heroes include bacteria, fungi, and algae. Each with unique strengths, bacteria offer immense metabolic diversity and rapid growth. Fungi excel at breaking down complex molecules with powerful enzymes, and algae help remove heavy metals and excess nutrients through biosorption and bioaccumulation, working together in many remediation systems. At the microscopic level, specialized bacteria and other microbes actively metabolize pollutants, breaking oil and organic contaminants into harmless byproducts. This slide highlights how microbial metabolism turns environmental threats into energy and biomass, essentially letting nature's tiniest workers do the heavy lifting in cleanup efforts. Bioremediation works through three main mechanisms: degradation, where enzymes break complex pollutants into safer compounds; sequestration, where contaminants are trapped or immobilized in cells or soils, reducing mobility; and detoxification, where harmful chemicals are transformed into less toxic or non-toxic forms by microbial metabolic pathways. The effectiveness of microbial cleanup hinges on environmental factors like oxygen availability, moisture, and temperature, plus chemical conditions such as nutrient levels, pH, and contaminant concentration. Managing these variables is key to encouraging the right microbes to thrive and carry out efficient degradation at contaminated sites. Chapter 3 focuses on practical methods and real-world applications of bioremediation. We'll compare on-site and off-site approaches, discuss engineered systems like biopiles, and walk through case studies that show how biological solutions are applied in controlled and field environments. In situ, bioremediation treats pollution where it exists, avoiding excavation and reducing costs and disruption. Techniques like bioaugmentation add specific microbes, while bioventing and biosparging boost oxygen to stimulate degradation, though the approach can be slower, and effectiveness may be limited by soil compaction and heterogeneity. Ex situ bioremediation brings contaminated material to controlled environments for faster, more uniform treatment. Common methods include land farming, composting, biopiles, and slurry phase bioreactors, offering better monitoring and quicker timelines, though they require excavation and transport of contaminated soils. The Santa Susana Field Laboratory project illustrates how targeted bioremediation can work. Local microbes capable of degrading PCBs, PAHs, and heavy metals were identified. Then nutrient addition and microbial enhancement sped up degradation, achieving significant contaminant reduction while avoiding costly excavation and disposal. Biopiles are engineered heaps that combine microbial processes with forced aeration, moisture control, and monitoring systems to accelerate pollutant breakdown. By optimizing conditions within these piles, biopile systems dramatically shorten cleanup times while keeping treatment contained and manageable. Emerging contaminants like plastics, pharmaceuticals, and microplastics require advanced strategies. Innovations include genetically engineered microbes with improved degradation pathways, nanoparticle-enhanced remediation to boost microbial access and activity, and carefully designed microbial consortia that create synergistic breakdown of complex pollutants. Bioremediation offers many advantages. It's eco-friendly, cost-effective, minimally disruptive, and can restore ecosystem functions with little secondary waste. Yet, it has limitations too. Remediation can be slow, site-specific factors affect performance, careful, long-term monitoring is required, and some pollutants remain resistant to biological degradation. Chapter 4 looks to the future, exploring innovations and persistent challenges. As research progresses in microbiology, genetics, and engineering, bioremediation's reach and efficiency are expanding, but success will depend on integrating technology with sound policy and community partnerships. Integrating bioremediation with green technologies multiplies benefits. Phytoremediation uses plants and root microbe partnerships. Nanoremediation leverages particles to increase bioavailability and reaction rates. And digital monitoring coupled with AI enables real-time optimization of conditions to maximize cleanup efficiency. Policy, regulation, and social acceptance are critical to scaling biological cleanup. Supportive regulatory frameworks, active community engagement, and transparent public education build trust and ensure projects meet both technical and social needs, creating the collaborative conditions necessary for long-term success. Bioremediation has global potential. Millions of contaminated acres could be reclaimed biologically. Documented projects show substantial cost savings compared with traditional methods. And these approaches align with UN Sustainable Development Goals by restoring land and water while reducing hazardous waste. Transforming a toxic wasteland into a vibrant ecosystem is possible through patient application of bioremediation techniques. Given time and the right biological tools, severely contaminated sites can be rehabilitated into healthy habitats that support diverse plant and animal life, showcasing nature's resilience when guided thoughtfully. In conclusion, bioremediation harnesses nature's own tools to address pollution, and continued innovation will broaden its impact. By combining scientific research, supportive policy, and community stewardship, we can restore degraded environments and move toward a sustainable future, one microbial process at a time.